Hello friends, welcome to Sam online class. Friends, today we shall discuss about the quaternary structure of protein. There are many globular proteins which are composed of two or more than two polypeptide chains. These polypeptide chains are called as subunits, and the proteins that are composed of two or more than two peptide chains are known as oligomeric protein. In oligomeric protein, one subunit associate with other subunit by the help of different type of linkages like hydrogen bond, disulfide bond, ionic bond and hydrophobic bond that give rise to another level of organization that is known as quaternary structure. So quaternary structure is a specific arrangement of subunits in a oligomeric protein. The quaternary structure of protein also refers to the spatial arrangement of subunit and the nature of their contact. So the quaternary structure is the arrangement of individual subunits in a oligomeric protein in a specific manner so that a compact structure can be formed. As an example of protein quaternary structure, we will discuss about the structure of hemoglobin. Hemoglobin is a tetrameric protein. Its main function is to transport oxygen. And hemoglobin, there are different forms of hemoglobin. All are composed of 4 polypeptide chains. The human hemoglobin exists in different forms. They are embryonic hemoglobin, fetal hemoglobin and adult hemoglobin. There are different forms of human embryonic hemoglobin. The human embryonic hemoglobins were discovered in 1961. The primary embryonic human hemoglobin is HB-Goer1. The hemoglobin Goer1 is composed of 2 zeta chain and 2 epsilon chain. Another embryonic hemoglobin is expressed at low level. That hemoglobin is hemoglobin Goer2. Hemoglobin Goer2 is expressed at low level and is composed of 2 alpha chain and 2 epsilon chain. There are some minor forms of embryonic hemoglobin. One is hemoglobin Portland 1. Hemoglobin Portland 1 consists of 2 zeta chain and 2 gamma chain, and hemoglobin Portland 1 is expressed at low level during embryonic and fetal stage. Another minor hemoglobin is hemoglobin Portland 2. Hemoglobin Portland 2 is also expressed at low level during embryonic and fetal life, and it consists of 2 zeta chain and 2 beta chain. The human embryonic hemoglobin are produced in the blood islands in the human embryonic yolk sac in the mesoblastic stage. The mesoblastic stage starts from the 3rd week of pregnancy up to 3 months. So there are various forms of hemoglobin. Another form of hemoglobin is fetal hemoglobin. Fetal hemoglobin is the principal hemoglobin in the fetal stage, and the fetal hemoglobin consists of 2 alpha chain and 2 gamma chain. It is the principal oxygen carrier in fetus. There is adult hemoglobin also. The principal adult hemoglobin is HbA. HbA is the principal hemoglobin in human adult, and it is composed of 2 alpha chain and 2 beta chain. There is a minor hemoglobin in adult known as hemoglobin A2. Hemoglobin A2 is composed of 2 alpha chain and 2 delta chain. All these different forms of hemoglobin are composed of 4 polypeptide chains. Alpha chain and zeta chain are composed of 141 amino acids, whereas beta chain, gamma chain and delta chain are composed of 146 amino acids. Hemoglobin is nearly spherical and its diameter is about 55 angstrom. The three-dimensional structure of horse adult hemoglobin was elucidated by Max Perez and John C. Kendrew and their colleagues in 1959. Hemoglobin A is the principal hemoglobin in adult. HbA is composed of 2 alpha chain and 2 beta chain. The alpha chain contain 141 amino acid. In this chain, valine is the N-terminal residue and arginine is the C-terminal residue, whereas the beta chain contain 146 amino acid. In beta chain, valine is the N-terminal residue and histidine is the C-terminal residue. So in hemoglobin, totally there are 574 amino acids. This structure explains about the quaternary structure of hemoglobin. In this quaternary structure, there are 2 alpha chain and 2 beta chain. Each alpha chain and each beta chain has its own tertiary structure. In alpha chain and beta chain, there are several alpha helical segments that are separated by bands. So these 2 alpha chain and 2 beta chains interact in a specific manner with the help of disulfide bond, hydrogen bond, hydrophobic bond and ionic bond to form a complex three-dimensional structure that is known as the quaternary structure. In this diagram, you can see that each subunit is associated with a heme. This red color structure is the heme, and heme is an iron containing prosthetic group which can bind with molecular oxygen. The heme rests in a crevice in this individual subunit. The heme has an iron atom at its center. The iron atom, which is present at the center of the heme, has 6 coordination sites. Out of 6 coordination sites, 4 are occupied by the nitrogens of tetrapyrrole ring and remaining 2, one is occupied by the R group of histidine and the remaining coordination site is occupied by molecular oxygen. So friends, in hemoglobin, 2 alpha chain and 2 beta chain associate and interact with the help of different type of linkages to form a complex three-dimensional structure that is known as the quaternary structure. For more updates, please subscribe my YouTube channel Sam Online Class. In next video, we shall discuss about the structure and function of plasma membrane. Thank you very much.